Happy Monday, everyone. You don't have to worry, I'm not going to be singing, so you can't really follow and act like that. So I'm going to just get into what we need to worry about for your final exam. I do want you, though, to really take good care of yourselves over the next little while. So after Wednesday, I'm not going to see you on a regular basis, which means that you'll be off studying or doing exams, and it's really hard to keep track of like where everyone is in terms of your mental health, your physical well-being, and everything else. But I do hope that you're going to take very good care of each other as well as yourself over the next little while. So we're going to jump back into Alzheimer's disease. We're going to talk about sort of the last little module that we need to think about. This is actually a really fascinating field right now. We're seeing Alzheimer's disease and dementia kind of evolving and changing over the last 2 to 3 years. It's not the same as it was 2 to 3 years ago, and the field itself is moving so quickly and rapidly, and it's changing in ways that no one would have predicted that it was going to change. And some of that's being driven by genomics, some of that's being driven by artificial intelligence, some of that is really just being driven by the fact that neuroscience is now incorporating more and more immunology into what it's studying. And we're going to come back and review a lot of those different aspects today, and also get you ready for your final exam. Before I get there, I need you to start thinking about how you're going to be preparing for your final week that we have together in class, and how you might start thinking about what you need to do and what you need to be able to finish over the next little while. And some of it is really quite easy to make sure that you are finishing things like your quizzes, your quiz 4 and 5. About 5% of you have not done it yet. You're kind of waiting. I'm not sure if you have other things this week. I'm told there are lots of term tests and essays and other things that are done and due this week. So please make sure you're staying on top of that. That is a pretty hard deadline. You've had like a couple of weeks now, and that time is actually running out for you. So make sure that you are doing that this week. Start preparing as early as possible for the final exam. And I don't like giving sort of this broad-based and sort of nebulous and not very coherent get ready for the final exam, because you hear that in every single class, and that's not very helpful. So what I mean by starting to get ready for your final exam, you should really make sure that your notes are up to date. So that's one. Make sure your notes are up to date. Number two, make sure that if your notes are not up to date, that you have access to a recording or to something else that you can get it up to date. And then three, when you start preparing your notes, making sure that they're all up to date, figure out which ones are going to be more troublesome for you. Like which ones did you have problems with when you were listening in class, and then maybe start to make an appointment to come in and see me over the next little while. So that's what I mean by preparing early. Do that when you have the time, in between lectures, just making sure that your notes are up to date and that you're ready for what's coming up next. So Wednesday, I've been asked to make it more of a review. So everything that we cover up to today will be on your final exam. Wednesday will be kind of a review to make sure that you're ready for your final exam and what to prepare specifically, like what sections, how to study from the different lectures. There are a lot of lectures now if you've already started, and how you might get ready specifically for each individual section. So the other thing that I was asked repeatedly, you don't have to ask. The reweighting will be done automatically. We'll take a look at term test 1 and term test 2. Whatever's higher for you, we will automatically reweigh it to be worth more. And the one that you didn't do quite as well on, we'll reweigh it to be slightly less than what was originally in the course syllabus. So there will be also coming up, because as I said, most of you will actually not start to come down to campus soon. And there will be a couple of online documents. Those of you who have had me for neurogenomics know that I like using things like Padlet. You can embed videos and other things. And it's pretty self-explanatory, but I'm going to have these online documents that will be ready for you starting on Thursday of this week, so that you can start getting ready, start understanding what you need to know. And then we will also have an online annotation document called Hypothesis, where you can actually write on top of different worksheets that I'll be posting for you as well. It's really trying to get you ready as best as you can for the final exam over the next 2 weeks, because your exam is about 2 weeks off. So some of you are also wondering about the announcement. What does it mean when he says that we will talk about how to adjust your final marks? So if you are thinking that I'm going to be adjusting your final marks down, you guessed incorrectly. I'm not trying to lower your marks by any means. And so usually in the past within HMB or within CJH, we have, up until like a year ago, done an automatic rounding to the next GPA bracket. So if you had an 89, we would used to actually round up to a 90. You didn't have to ask. You didn't have to go and get the different exam remarking sheets, et cetera. And so I think because it's been a really good semester, I've really enjoyed teaching you, we're probably just going to go ahead and reinstitute that so that if you are sitting at a 79, we will automatically round you to an 80. If you're at an 84, we will automatically round you up to an 85. And an 89 will automatically round you up to a 90. And that's what I meant by going back old school to what we used to do. So I'm hoping that's okay with you. If you have like really strong objections, don't do this, then you can let me know after class. And I'm hoping that most of you don't have an objection with that. So here we are talking about Alzheimer's disease, the destruction of synapses. And you've probably heard a lot about Alzheimer's disease, and you probably have thought a lot about Alzheimer's disease. And here is where we're at. We're actually no better off than where we're at with stroke. So it's not exactly that we've tried 700 different drugs for Alzheimer's disease, because Alzheimer's disease is a long process. It's not something that you pick up immediately after blockage of a blood vessel, for example. But many, many, many drugs have been tried, and every single one of the drugs that have in theory worked out so well in animal models, every last one of them, including last week, there was a drug that was in phase 2 of clinical trials. It also failed at that particular trial. This was a fairly big trial that was being done in Japan. A lot of hope. There was a lot of great hypothesis testing, but it simply didn't work. And so we're right back to where we began. Nothing. We have 0 drugs. We have 0 therapies. We have nothing to actually help someone who has Alzheimer's disease. And here's the kicker. We know a lot about the disorder, and there are lots of things that you've heard about, but I want you to think about what you know and what you might rethink a little bit in terms of what you have heard about Alzheimer's disease. And so here's where we're sitting right now, in this sort of place where we're looking at the last lecture related to synapses and why synapses might not be the be-all and end-all. So knowing these different models is really important. That's what we've been emphasizing throughout this semester, that sometimes these models are very, very useful. And sometimes these models are not so useful for a variety of reasons. And at least for stroke, and at least for Alzheimer's disease, I'm going to be showing you that these models and these different approaches have not been very, very useful. And we have to rethink how we're approaching Alzheimer's disease if you are going to believe the statistics that I'm going to show you in a few moments. Before I get there, I also wanted to take a little bit of a sidetrack, and you don't need to worry about the details on this slide per se, and you don't need to worry about taking down notes on this slide. It's really to give you a little bit of context. And some of you will have had lectures with me, for example, on the gut microbiome in 3rd year, and why the microbiome is sort of an area of interest that people are looking at. And one of the things that I wanted to tell you is Robert Moyer and Rudolf Tanzi actually had this really interesting hypothesis that we're actually going to revisit later on today, and to give you an idea why some of these ideas in Alzheimer's disease research are actually coming back to the forefront. If you are going to look at this molecule that we're going to spend a lot of time on today, and I'm going to tell you that you need to know this pathway that we're going to talk about related to amyloid, because it's still the dominant hypothesis, and we've talked about beta amyloid and amyloid precursor proteins and lipid rafts way, way back in the beginning of the semester. If you look at beta amyloid, this sort of hallmark feature of Alzheimer's disease, it kind of looks a little bit, and there's a lot of similarity, and there's a little bit of hypothesis testing that Robert Moyer and Rudolf Tanzi, who's an expert in Alzheimer's disease, one of the things that they looked at is, hey, you know, this kind of looks a lot like an antimicrobial protein that we would often see associated with bacterial infections of the brain. And this is actually now sort of a new field that people are interested in. And you might be wondering, why bacteria and why the brain? Isn't there this blood-brain barrier that we keep hearing and talking about so often? And isn't this working in Alzheimer's disease, or is it not working in Alzheimer's disease? And that's really the question. We don't know. And that's also one of the things that I'm going to propose to you. So this interesting theory that they developed, and there's some evidence to support this, is that some of that Alzheimer's disease protein that we're going to be talking about, that amyloid beta, the beta amyloid that we have heard a lot about in Alzheimer's disease, actually resembles things that are related to bacteria, antimicrobial proteins, bacterial membrane walls. Here they're looking at different candida molecules, or molecules from candida, but ultimately they feel very strongly, based on their studies, that when they look at individuals that have Alzheimer's disease, that they actually see a lot of this LL37, which is related to beta amyloid, related to bacterial cell walls, in the brains of individuals that have Alzheimer's disease. That's curious. It's kind of fascinating if you think about that. How did it get there? Is it a cause? Is it a correlation? Or is it just a fluke? Because is it the same fluke that all of you have been exposed to in the past? So for example, how many of you have heard something about Alzheimer's disease, that if you use aluminum pots or use aluminum pans, that you can actually increase the incidence of Alzheimer's disease? Have any of you heard that theory? This is an older theory, and it turned out to be wrong. But for a very long period of time, people thought that Alzheimer's disease was caused by cooking with aluminum-based cookingware. And that's because our ideas and the research data actually was faulty. And here we need to understand, is this research data, where you're finding bacteria in the brain of people with Alzheimer's disease and at higher concentrations, is this faulty data or is this new data? And we always need to go back and examine data that way. So here we're going to look today a little bit more at Alzheimer's disease. We're going to go back over some of the gross pathology, and there are parts of this gross pathology that I need you to be very clear and to understand and to think about. In fact, it's outlined for you in your lecture notes to start thinking about that today after lecture, so that you won't be surprised by things on your final exam. So let's talk about that a little bit later on. And the vast majority of what we are going to talk about today is about beta amyloid, this sort of mysterious, what does it do? Why is it so toxic to neurons? How does it actually work? Why does it get released in an activity-dependent manner? So neurons are more active, they release more beta amyloid. But you've been taught over and over again in almost every lecture or every sort of news article that doing puzzles and cognitive thinking actually enhances your ability to ward off Alzheimer's. But I'm telling you that the research is very clear. The more active your neurons are, the more of this beta amyloid it releases. And if beta amyloid is bad, then maybe you shouldn't be doing puzzles, which is not true, by the way, but it gets you to start thinking a little bit about what we know and what we don't know about this disorder. And then we'll talk a little bit about this link with cholesterol and why this cholesterol molecule is important for late onset forms of Alzheimer's. So the next few slides are ones that you don't need to worry about at all. And you've all heard the news that Alzheimer's disease, just like stroke, is going to be increasingly important within your era. So when you're off and doing clinical practice or doing research, or when you're starting your families, and when you're taking care of your parents who are getting older, and you start having to worry about Alzheimer's disease, it's going to be impacting you within about 10 or 15 years. And think about that. So you're about to face the burden that we weren't able to address. So when I was studying at the Center for Research in Neurodegenerative Diseases, our mandate, our goal that was put out by the Society for Neuroscience was that by the end of the 2000s, we will actually have understood everything about the brain. Obviously, that hasn't happened. And so we're kind of leaving it up to you to kind of address this growing amount of Alzheimer's disease burden within North America, within the Western Hemisphere. And will it be on the lower end or the higher end? We're still not sure. So for a while, it kind of looked like we were going to be on the lower end in terms of the overall numbers by about 2020, not that far off, just 1 year. And now it kind of looks like maybe we're going to be a little bit above that level. But ultimately, when you're looking at what you're going to be doing 10 years from now, you're looking at this bigger number down here at 2030. And if you think about how drugs work and how much testing has to go in, and I've told you that there are no drugs right now in phase 2 clinical trials anywhere, then we don't have a lot of options. And so as individuals that are going to be facing this increased burden of having more Alzheimer's disease, this is something that all of you, whether you stay in neuroscience or whether you don't, actually should be considering. So all of you will know things that the brain is changing. The synapses are being lost. These synapses are no longer as healthy as they once were. The normal synaptic transmission, all these different synapses, glutamatergic, cholinergic synapses, most of the synapses within the brain are going to be affected. And so almost every system that you think about, whether it's related to memory directly or indirectly through being able to pay attention, all of these things are going to be affected. So not surprisingly that memory, attention are the two top symptoms, but they're not by any means the only symptoms. We also have, as different areas of the brain are going to be affected, changes in mood, changes in your ability to emotionally process and regulate, your levels of anxiety, depression, and other things are all going to be changing as well. Your ability to make good judgments are also going to be affected. And not surprising if you think about all these different systems that are being affected within Alzheimer's, that as every single synapse, if we believe this model to be true, is going to be affected in this Alzheimer's disease model. And again, is there increased anxiety with individuals as darkness or as evening approaches? Also a really interesting hypothesis about whether or not circadian rhythms, your core circuit. So think about the hypothalamus. Your hypothalamus is there to make sure and ensure that all of your core processes are going to be maintained. But if your hypothalamus, which is part of that circadian rhythm, is being affected, how much of your brain can be spared if you're thinking a little bit about this. So again, without going into a lot of details, this slide is just there for you to make some context and to understand the overall big picture of the issue that we're facing, is that if we're looking at the loss of neurons and therefore the synapses and the connectivity between different groups of synapses, not surprisingly, there's a disease process that we can identify. We can see that we have amyloid plaques consisting of these different fibrils or groupings or clumpings of beta amyloid. And then inside the neuron, we will also have hyperphosphorylated tau, all summarized again for you later on just to give you some perspective. And we know that there are certain areas of the brain that begin in many individuals that affect memory, attention, judgment, etc. So areas of your medial temporal lobe that house your hippocampus, the amygdala, and other associated structures, they tend to be the first affected, but they're by no means the only areas of the brain that are affected. And all of these, again, will come into play in a few moments. So this is an important slide. So here's where you need to start paying a little bit more attention. And here's where you also need to start thinking. And you need to start thinking about things for your final exam to make sure that, one, this doesn't come out of the blue, that you realize that this is something to think about and to go back over your notes when you have the time over the next 2 weeks before the final exam to get ready for those types of questions. So we know that the loss of brain tissue, so whether the neurons are shrinking or we're losing these neurons, either way, brain atrophy or loss of neurons, starts in areas like the medial temporal lobe that house the hippocampus, but then it spreads, right? And that becomes an interesting idea. So why does it look like it's spreading? And this actually is the sort of underlying hypothesis behind sort of modern neuroscience. Can these molecules, if you have beta amyloid in your brain, if it were somehow transmissible, if you could take it out of your brain, can you actually cause it to spread in someone else's brain? And there's a lot of evidence to suggest that these misfolded proteins, whether they're prions or beta amyloid or hyperphosphorylated tau, all of them are misfolded. They're aggregates, they're clumps. They shouldn't be in clumps, but they are. If you take those clumps and put them into a normal brain, they too will clump in your brain. And that's sort of the modern hypothesis. And this goes along with how this is spreading from one area of the brain to the associated areas of the brain, because that's how the brain is mapped out. It's the primary cortex, the association cortices around it, and it spreads from that one area to the next area. So again, fascinating, interesting, and very curious how this disease tends to spread. One area that becomes very important, and this goes back to attention. If you have taken a physiology course in the last little while and you've learned a little bit about the cholinergic projection fibers, one of these nuclei is known as the nucleus basalis of Maynard. And this nucleus basalis of Maynard is one of the key cholinergic nuclei, so groups of neurons that synthesize and then release choline at all of these different areas. In fact, all of the cortex is innervated by the nucleus basalis. So this is releasing acetylcholine to all of these different areas. Now, how do I need you to think about this? And what does it mean by how to target this area? So we know a couple of things. We know that neurons can be identified by the types of neurotransmitters that they produce, and that these types of neurotransmitters that are produced, things like dopamine, for example, often have a synthetic enzyme that's associated with it. Now, if you've taken other neuroscience courses, or you happen to have studied, or you happen to remember, there is a synthetic enzyme that we've talked about related to acetylcholine. And you might want to think about that. You might also want to think and relate this back to the fact that one really specific targeting system that we looked at in terms of molecular genetics actually subverts the system. So think back to muscarinic receptors, and have we talked about the molecular biology and the alteration and the adaptation of muscarinic receptors in this course, and how might that be useful or not useful in getting ready to think about how to subvert this cholinergic system on the final exam. And you can see that it spreads to a number of different areas across the entire cortex, into areas of the amygdala, hippocampus, entorhinal cortices, etc. So it has wide-ranging connections, and this area of the brain is also subject to loss after the increase in beta amyloid. So again, we get this loss of neurons, and of course the loss of these synapses, and we get the loss of specific synapses. The ones that are cholinergic tend to be also widespread, but also large in number in terms of the losses. And eventually, we will see within the brain, and this again is a cause of great, great controversy. Is it the beginning, or is it actually the end result? Is it the cause, or is it just something that happens as a byproduct? So does beta amyloid actually cause neurons to die? Well, every single therapeutic approach that we have taken so far hasn't worked. And you have to wonder, if it isn't the cause and it hasn't worked, maybe we're looking in the wrong place. And that first sort of headline that I showed you is that some individuals actually believe that beta amyloid is just sort of a byproduct of what happens to the damage in neurons. And in fact, we should be looking at these neurofibrillary tangles first. And this is sort of the debate. It's literally split down the middle. Neuroscientists either like beta amyloid or the neurofibrillary tangle hypothesis, but it's split in half. And so when you have science sort of split in half, it doesn't really go anywhere. It doesn't become fruitful or meaningful, and we have to think about what that means for us in 2030 and in 2040, etc. So we know that there is lots of glial scarring. So think back to what we talked about earlier in this course, not that long ago, about how we have these parenchymal glial progenitor cells sitting in these different areas of the brain, ready to swoop in to areas that have undergone damage. And now damage is occurring all across the brain. What would that mean for glial progenitors dividing, and how they would be migrating in terms of the glial scarring that would occur? And of course, the most interesting one that we tend to not talk about, which we should be talking about, which is this intersection between neuroscience and immunology, which is the immune response and the microglial cells. And this is actually probably the way in which we should be approaching Alzheimer's disease, but to be honest, most neuroscientists do not have a strong immunology background. And so we tend to not worry too much about this pathway. And so we need you to start thinking about joining immunology and neuroscience together in terms of therapeutic approaches. Many of you will know about cancer and immunology, but neuroscience and immunology is sort of the big hope that we're all kind of aspiring to. So this slide is one that gives you an overview of where we're going to be going next, and you do need to know these next few slides in a lot of detail. So here's where you start circling off that you need to go back and review these different types of processes. So in all neurodegenerative diseases, every single one that you look at, every last neurodegenerative disease, it does not matter what you are looking at, whether it's genetic in origin or whether it's something that we don't know yet. Alzheimer's is a good case of this. We always see some kind of aggregating misfolded protein. And here, beta amyloid is one of the misfolded aggregating proteins, and we develop these amyloid plaques that we can stain the brain for and go back and identify. And these extracellular aggregates of beta amyloid or amyloid beta are the ones that we think, and again, there's some evidence, strong evidence, to suggest that if you have more and more of this beta amyloid around the outside of a neuron, you can actually induce cytotoxicity. Those neurons start to die dependent on the concentration of the beta amyloid that is around outside of the neurons. Interestingly, and very curious, the more activity that you have, the more action potentials a neuron fires, the more of these aggregates we actually start to form. And again, it becomes a really interesting sort of corollary to what we think about in terms of action potentials and cytotoxicity. So we'll come back to that a little bit later on. And one of the things that I've mentioned to you previously, like early on in the course, is the fact that we have amyloid precursor protein. All of us have amyloid precursor protein. And we have this is an integral membrane protein, so it's got a transmembrane domain, it has this large extracellular region. It serves a number of different functions. We need it developmentally, and yet something happens to this during times when you get older, or if you have a genetic mutation that cause this to be processed into two different pathways. One, which will form a non-aggregating form. So this doesn't clump, this does not cause toxicity, and when you process it using alpha and gamma secretases, which I'll show you in a moment, we form this non-amyloid pathway. And that's better for your brain. Your brain can clear these non-amyloid, not aggregates, but these non-amyloid fibrils more easily. And then there's this amyloidogenic pathway, and this involves beta secretase, and that's where the name beta amyloid comes from. That the fact that amyloid precursor protein is cut by beta secretase that causes beta amyloid to be formed. So again, I know you have this slide in a slightly different way. There's a lot on this slide, and to be quite honest, you need to know this slide for your final exam. There are some short answer questions related to knowing the different details on this slide, as well as multiple choice test questions that you're likely going to be asked based on this slide. And so if you know that you need a little bit more time to try to process the information and make meaning of that information, then this slide is one you should probably think about going over when you can. So this non-amyloid pathway, and this is sort of the amyloid precursor protein. All of us have it. Our neurons are rich in amyloid precursor protein. And we have lots of this amyloid precursor protein, and it can be cleaved in different ways. So it can be cleaved either by an alpha secretase going toward the right, toward this non-amyloidogenic pathway, or it can be cleaved by beta secretase, which takes you to the left, and that's sort of the not so good pathway. And we can tell by examining different fragments that we can detect either in the cerebral spinal fluid or in other compartments, which of these pathways is dominating. And many of the different types of therapies that have been tried are to enhance the activity of beta secretase and to enhance the activity of the alpha secretase and to block or inhibit the pathway of the beta secretase. And those haven't worked. They are not good trials. They've stopped for a variety of different reasons. But along this pathway, as we get alpha secretase in green sort of being activated and this amyloid precursor protein is being cut by this cysteine protease, so this is a cysteine protease. That's going to be important in a few minutes. And the cysteine protease is going to cleave this into two different fractions. One is this soluble amyloid precursor protein alpha, as well as a smaller intracellular fragment known as CTF83. And we know that, therefore, if we detect either of these, that the non-amyloidogenic pathway is going to be activated. And then this other gamma secretase that we'll talk about a little bit later comes along and takes this fragment here and will further cleave this into a P3 and an AICD, or an A-beta intracellular domain. And neither of these is toxic. And so we have this way of taking this large molecule and converting it or breaking it down into a pathway that's not harmful at all for your brain and can be cleared very easily. And if you're thinking about this, and maybe start thinking about this, when we talk about clearing these molecules, these proteins, and these fragments from your brain, what cell types do you think might be involved in that? Guesses, ideas, suggestions? So microglia would be a really good source, and maybe astrocytes might be important as well, but we need to start thinking about the whole picture now to kind of start wrapping up the course in terms of what you now should be able to tell me. So the other pathway that seems to be very toxic is this one that actually forms this amyloid beta. And this is where we get the beta secretase being involved. And this beta secretase will cleave this amyloid precursor protein at a slightly different point in the precursor protein. And it will actually form a 99 amino acid long chain. And this is that CTF99 form that you see here, and then this other soluble amyloid precursor protein beta, a smaller fragment than the one that you see here for alpha. So you can tell that the size is a little different, but they're soluble, which means that they can be cleared or removed from the brain fairly easily through a number of different mechanisms that have been identified to be able to clear both the APP alpha and the APP beta. But the problem is that now, after this beta secretase has caused this cleavage, that we now have the activation of the gamma secretase. And its job is to come in and clean up these small little resulting fragments. The issue here, though, is this gamma secretase, when it now does its job and it cleaves at this interface that you see here, it will actually cleave off a slightly bigger fragment than you would have over here on the right. Fragment, this amyloid beta, seems to have the ability to be able to clump. It makes connections with itself. It starts to form dimers and then oligomers, and then these oligomers start to form larger clumps. And these clumps are very, very toxic to the brain. And we know that this process, this entire process, whether it's to the left or to the right, can actually be accelerated with increased activity within the neurons. The more active the neurons are, the more action potentials the neuron fires, both pathways, including the amyloid pathway, will be enhanced. These enzymes are also going to be more active. And as I mentioned here, some of these different fragments, like the AICD, can actually be degraded, similar to what we saw on the right-hand side of this diagram. But these other A-beta forms are not. And we have known that for a very, very long period of time. In fact, the fragment within the A-beta 1 to 40 and the A-beta 1 to 42, the amino acid sequences between 25 and 35 are the ones that seem to be the most toxic out of the entire fragment. So that's like one hypothesis, that amyloid beta, in response to increased activity, gets released and it starts to actually get processed. And as it gets processed, it will actually cause the loss of neurons. And we'll come back and revisit that in a few minutes. So the other pathway that is also gaining traction and gaining a lot of popularity, mainly because all of these other clinical trials, the one last week in Japan and the one 2 months ago that was occurring down in the United States, all of these have kind of been targeting amyloid beta, and they haven't been really working. And so people are also interested in maybe before amyloid beta happens, there's something wrong with the transport mechanism within a neuron. And maybe this means that this tau protein, which links these microtubules, so it's a microtubule-associated protein, and this microtubule-associated protein is somehow not able to connect with these microtubules, and that means that the microtubules are no longer stable. And these tau proteins, which are involved in their stabilization, can be phosphorylated. There are a number of phosphorylation sites that are available, and as you phosphorylate tau, it makes it less and less likely to bind to microtubules. So that means the microtubules are at risk, and those axons that we looked at earlier, these axons, these long axons, the reason why they're long is that we get these long microtubules that are found within them. And they're useful for transporting different organelles and different resources from the cell body to the axon, boutons. And we know that if we disrupt that process, the neurons actually start to degenerate. So the increased levels of phosphorylated tau or phosphotau that form these intracellular aggregates that we can stain in the brain actually also suggests that this process is important. So this process might also be important in Alzheimer's disease as well. And again, not surprisingly, when we go in and we start to look at phosphorylated tau, when we look at an Alzheimer's diseased brain and it has a lot of progression of the disease, so the intensity or the duration of the disease is longer, we see more and more of this hyperphosphorylated tau, suggesting that it's accumulating. And it's just a suggestion, and there's just a correlation. There's no direct causation, but it suggests that these neurons are indeed in the process of dying. And so, again, another hypothesis that so far, the fewer trials that have been tried haven't been really successful. So you'd have to kind of think a little bit about this, because I didn't tell you, nor do you have to know this for your final exam. But when we talk about phosphorylation, generally we are talking about kinases, and kinases add phosphoryl groups to proteins. And so inhibiting the specific kinases in different trials hasn't worked, nor has the opposite process. If you're thinking about it, dephosphorylating some of these phosphorylated tau might also reverse that process. That hasn't worked yet either. So again, we're kind of stuck in terms of what we have in terms of the different model systems. So I'm actually not going to ask you, to know all the details of the different Alzheimer's disease models. There are a number. In fact, 2 of these models originated here in Toronto. Toronto, back in the 1990s, through a lab at the Center for Research in Neurodegenerative Diseases with Dr. Peter St. George Hislop, actually identified a number of mutations in familial Alzheimer's disease, and they created transgenic animals that are used worldwide, and these are known as the TANS or CRND mice, and you don't need to worry about them. However, I wanted you to be aware of the fact that we have a number of different animal models, primarily mice, where these mice don't have issues with their beta amyloid. We have to introduce human amyloid into these mice brains, so it's a transgenic in that sense. And when we start doing that, or we start introducing mutations into these mice, some of these mice are better models for Alzheimer's disease than others. And there are, again, a number of them. And I'm mentioning the APP mouse up here for the time being, because one of the very first mutations that was found was in a small cohort of Swiss family members where they had a mutation that was identified through early genetic screening in APP, the amyloid precursor protein, that led to an enhanced formation of beta amyloid, and they developed in a genetic fashion earlier forms of Alzheimer's. So they put that mutation into a mouse genome. It got expressed in the brain, and these mice kind of look like they have Alzheimer's disease. And then other mutations in presenilin were identified, and other mutations in tau. But all of these are a little bit different. And what this is actually showing is that when we look at the mouse brain, so for example, APP23, the mouse model that I just indicated to you, if we do either Congo red birefringence, where we stick a molecule that binds only to beta amyloid, we'll be able to see this type of pattern, which looks very similar to the disease brain neurons that also have beta amyloid. And you can see they kind of look similar. And if we look at the mouse models and the human brain for neurofibrillary tangles, depending on the mouse model, we'll also see very similar patterns of staining for neurofibrillary tangles. And so you kind of wonder, well, okay, get all of that, get the fact that we have different animal models, and you have to be very careful in how you interpret these different types of results, because sometimes the amygdala is affected, sometimes the hippocampus is affected, and sometimes it takes a long time for you to see these effects. But ultimately, even though we're looking at modeling human amyloid precursor protein, we are not successful. And these mouse models have a lot of limitations that are now being identified. So again, just before we think a little bit more, and there are a couple of slides coming up to get us to think about this. So if you look at Alzheimer's disease in general, the vast majority of Alzheimer's disease is actually for unknown causes. They are sporadic. We don't know what they're caused by. We have identified only a very small number of genes that can be directly implicated in Alzheimer's, about 5%. 95% of cases of Alzheimer's, if they are properly diagnosed, and we talked about how it has to be properly diagnosed, accounts for 95%. We don't know what the cause is. And so that makes us wonder, like, is there like a bacterial infection that causes Alzheimer's disease? Or is there a gene that we have not yet found that exists out there? Again, people want to know, like, what about that 95%? Only a small proportion, about 5%, have an identified gene. And out of that 5%, the vast majority of this that have this gene actually have very, very early onset forms of familial Alzheimer's disease, or FAD, as is shown here. And they get it much earlier, and it's much more prevalent in these individuals as well. Ultimately, though, the overall symptoms, memory loss, emotional changes, lack of judgment, all of those things seem to be identical between the genetic and the sporadic forms. So there must be some kind of link between these two different disorders. And of all of these familial Alzheimer's disease forms, so only 5% of all cases, but if we were to take a look at that 5%, at least 2 individuals would have a mutation in either presenilin 1 out of like 100 that we checked, or presenilin 2. And we're going to go over presenilin 1 and presenilin 2 in a few moments, or, as I mentioned, with the mouse models, APP. So before we get there, and again, you don't need to worry about all of the details here. There are a couple of things that I want you to be aware of. And this kind of goes back to what we're linking together. And this paper came out on Friday, so as I was kind of thinking, what should I talk about on Monday with this group? One of the things that we know now with genomics, we have unprecedented abilities to go in and start looking at mutations or single nucleotide polymorphisms that we have not been able to identify before. And this interesting paper that came out actually makes some interesting links that I'm hoping you will also kind of make some associations with. I don't know if you've ever heard of CADASIL, and this is cerebral autosomal dominant arteriopathy. It has some subcortical infarcts and, interestingly, changes in white blood cells, common. So in fact, the next example of this, Nasu-Hakola disease, also has leukodystrophies, also changes in white blood cells, and including some of those immune cells that we talked about earlier. So just keeping that in mind. So why is this Notch3 mutation interesting for us? This Notch3 mutation that is associated with this unusual form of Alzheimer's disease. So this is a genetically inherited form. They know that they're going to get Alzheimer's disease if CADASIL and Notch3 mutations are identified. These are actually interesting because, one, they start developing migraines, and guess what? In addition to that, they will also develop vascular changes. So you might have heard about vascular forms of dementia. That idea came about because of these individuals. These vascular forms of dementia where a Notch3 mutation has caused them to have strokes, mini strokes early in certain areas of the brain, ultimately leading to earlier forms of dementia is actually really interesting because it makes that link between stroke and Alzheimer's and maybe other disorders as well. The other disease and microglial cells. So keep in mind these white blood cells, microglial cells, the immune system are all being affected in both CADASIL as well as in Nasu-Hakola disease down here at the bottom. Nasu-Hakola disease is odd because your bones are actually being affected, and as your bones get affected, you get these little cysts and these nodules that develop as your bone gets remodeled. And you might think, okay, well, not terrible. But the other thing that's associated with Nasu-Hakola is the fact that you also get an early form of Alzheimer's disease. And the identified mutation here is TREM2. So for those of you who have taken IM250 or other immunology courses, you know that TREM2 is actually a really important regulatory molecule that's associated with a number of pathways in the immune system. And so we have to kind of go back and re-examine what we have thought about Alzheimer's disease in the past. If these two mutations that have been identified in these disorders that cause early forms of Alzheimer's disease that are impacting the immune system, but not directly neurons, we have to rethink and refocus like how we're going to approach in the future things like Alzheimer's. So one last thing, have you heard this headline? So you've heard probably back in early January, this paper came out January 23rd, that if you don't brush your teeth and you have chronic gingivitis, that you actually increase the odds that you'll get Alzheimer's disease. Did you see that headline? Anybody? Too busy studying? It was like term test 1, you didn't have time, January, couldn't do it. No one heard of this headline. No one. Surprised, I'm shocked. No one, really, no one? Wow, okay, well, all right, let me tell you. So there's this idea that if you don't brush your teeth and you have gingivitis, chronic swelling of your gums, that you actually have an increased likelihood of developing Alzheimer's disease. Going to go brush your teeth tonight? Do you brush your teeth at night? I'm hoping, yes? Okay, good. Brushing your teeth is really important. The fact that we used to have med school and dental schools kind of combined together, and we're now rethinking like maybe that's really important, is coming back into fashion. So if you didn't hear about this, you can look up these headlines. But I want you to keep this in mind. If you do see those headlines, go back and check out the actual paper. And you don't have this, but what this actually suggested was there's a particular type of bacteria that is very highly associated with Alzheimer's disease. And this type of bacteria is also very highly associated, just like Rudolf Tanzi was thinking earlier, also with like your gums. And if your gums bleed when you're brushing your teeth, it's likely that you have an infection in your mouth with this P. gingivalis. And this P. gingivalis, one of the interesting things about this gram-negative bacteria is it actually secretes this factor, which is known as a gingipain. And these gingipains, again, no big deal. Lots of bacteria secrete these gingipains. But when you look in the brains of people with Alzheimer's disease, there is a very, very high level of gingipains, which shouldn't be there, in the brains of people with Alzheimer's disease. The worse the Alzheimer's disease is, the more of these gingipains are actually found. And it, again, becomes really, really interesting when you start thinking a little bit about why people are looking at this. We don't have genetics, and so we need to identify the cause from a different biological origin. So I think now is probably a good time to take a break, go brush our teeth a little bit, maybe go use the facilities, and then come back in about 10 minutes. If you don't mind, we can just jump right into what we need to get to. Before I leave this slide, which is, for the most part, not something you need to worry too much about, I hope your laptop is okay. Okay, so I mentioned gingivitis, and I mentioned that if you have like chronic gingivitis or swelling of your gums, that there's a correlation between that and the development of Alzheimer's disease and the severity of bacterial infections, including like the P. gingivalis in the brain of individuals with Alzheimer's disease seems to be higher. But I also wanted to mention and draw your attention to the fact that these gingipains that are getting released by the bacteria that infect your gums and might infect your brain are also cysteine proteases. Have we heard of cysteine proteases before? Just a few minutes ago, and these cysteine proteases, which are outlined for you right here, these cysteine proteases are ones that these bacteria are normally acting to break down proteins that have these cysteine groups in a variety of different locations. And so the idea is that this could also work on the amyloid precursor protein because the secretases that I mentioned are also cysteine proteases as well. So again, the reason why we're looking in these unusual places that up until a few years ago no one thought of looking at is because we don't know all the causes for Alzheimer's disease, and we don't know all the reasons and the rationales behind how individuals develop that sporadic Alzheimer's disease. What causes it? Only 5% of the cases are actually identified, and we know a very, very small handful of genes, mutations in APP, mutations in presenilin 1, presenilin 2, which I'll get to in a moment. I've also mentioned Notch3 in passing with Cadicil, and then I've also mentioned TREM2, which is another mutation that is associated with an early onset form of Alzheimer's. And so when we look, overall, the genetics actually suggest that there might be something there, but we have only identified a very, very small subset of these different types of genes. And they're very, very rare when we take a look at them. So if you have mutations in APP, so of all of the identified, in case you're wondering, this 12%, how does that match up with the 5% that you mentioned earlier? Out of all of the identified genetically inherited forms of Alzheimer's disease, it makes up about 12% of all of those forms. It's not 12% of Alzheimer's disease, it's the inherited forms of Alzheimer's disease that we have a gene that's associated with them. So it's not the most dominant type of mutation, but these mutations have been identified with early onset forms of Alzheimer's disease. And again, one of the things, if you have taken a genomics course or you've taken and you remember what you learned in HMB 265 or your genetics courses, one of the things that we know makes this important is that in Down syndrome, on chromosome 21, there's actually an extra copy of the amyloid precursor protein. So increasing the load of amyloid precursor protein actually makes it more likely that you'll have breakdown products of amyloid precursor protein. And when you look at individuals that have Down syndrome, they actually have a lot of the features by the time they're in their late 30s, early 40s that resemble Alzheimer's disease within their brains. And the mutations that have been identified in APP or amyloid precursor protein make sense in the fact that we are going to increase the beta secretase pathways. You're going to break those down more frequently. You will increase the amount of the amyloid beta proteins or peptides more higher. And then this will also lead to the fact that I don't need you to memorize all of the details because I didn't cover it in a lot of details. When you look at amyloid beta, and the fragment is potentially toxic, there are two fragments, the 140 and the 142. One of them is more toxic than the other, and it tends to clump more easily than the other. And what this is saying is that the A beta 1 to 42 is the more toxic fragment. But again, that detail is not one that I covered in great detail here, so you don't need to worry as much about that. Just the overall picture of amyloid precursor proteins as a potential target for genetic mutations leading to Alzheimer's disease. The other one that I mentioned to you, specifically in your bulleted lecture notes, amyloid precursor protein was the first one, and then I mentioned that the other ones, about 50% of the genetic identified mutations involved the presenilins, presenilin 1 or presenilin 2. And these were, again, were identified here in Toronto in the late 1990s, early 2000s by Professor St. George Hislop and his team over at the Center for Research in Neurodegenerative Diseases, using a variety of different genetic approaches. And I wanted you, again, if it's been a while since you learned about this, or if you've never learned about this before, these presenilins are part of this larger complex. This complex that we've talked about are the gamma secretases. And these gamma secretases are actually important because they have that presenilin, either presenilin 1 or presenilin 2. They have another molecule known as nicastrin. They also have APH1, and they have this other molecule known as PEN2. All of that shown to you on the right in this diagram. That complex together makes up this gamma secretase. But what makes this an interesting molecule is only the presenilins actually cut proteins. And presenilins are the only part of this gamma secretase that have proteolytic activity. And one of the things that we know, based on Professor Hislop's work, is that if you have different mutations, and he identified about half a dozen mutations, and there have been many, many more that have been identified, that if you have mutations in presenilins, either presenilin 1 or presenilin 2, you actually lower the age of developing Alzheimer's-like symptoms in this form of Alzheimer's disease, or familial Alzheimer's disease. And again, the vast majority of mutations have been identified in presenilin 1. So 2 isoforms, but most of the mutations occur in presenilin 1. And what happens is this gamma secretase, because of the way that it actually processes APP, will actually lead to more and more production of the amyloid beta pathway, either the 40 to 42. Yes.
So if you remove presenilins, gamma secretase can't do anything?
So can you remove the gamma secretases completely?
No, if you remove the presenilins, they're the only ones with the proteolytic activity. That means gamma secretase can't do anything.
That's correct. So these are the only components that actually will cut the APP. Right. So since we're looking at the fact that we have a very, very small subset or proportion of genes that can actually explain Alzheimer's disease, and beyond the APP, beyond the presenilins, beyond all of those I also mentioned today, to make sure that you're all on the same page, NOTCH3 was another molecule, genetically identified molecule, found last week, but it seems to fit very well into the literature around immune system, dysfunction, etc. And TREM2, which was another one, also seems to fit in the genetics and is another identified genetic, potential genetic marker. So one of the other things that you might have heard of is that you have to worry about your ApoE status. And this is, again, related to cholesterol processing. And it's one of those interesting molecules that we need to be aware of. It's in the literature a lot, but it's also in popular media an awful lot in terms of Alzheimer's disease, late-stage Alzheimer's disease, and whether or not you are a carrier for different isoforms of ApoE or apolipoprotein E. And to date, still in 2019, if you are going to look at a risk factor, so we don't know 100% of what causes Alzheimer's disease, but if you're, again, looking at potential risk factors for developing stroke, we went through a number of these different risk factors for stroke, some modifiable, some not modifiable. One of the big risk factors that is non-modifiable, you can't change this, for developing Alzheimer's disease beyond sort of the familial forms is whether or not you have apolipoprotein E, what variant of this you actually have. And again, if we're taking a look, ApoE, apolipoprotein E, is normally produced by either the microglial cells or the astrocytes. And one of the things that it does, it is normally there to help solubilize different forms of cholesterol and allows for the processing and transport of cholesterol within your brain. And this is actually not trivial. If you think a little bit about this, if you look at white matter tracks, you look at how important myelin is, and you look at the overall contribution of cholesterol to the membranes, and you look at cholesterol to the membranes that contain lipid rafts and everything else, it's not surprising that cholesterol metabolism is really important within our brains. So if you look at the three major alleles that we have for ApoE that have been identified to date, ApoE2, if you have this, again, this is cysteine at these different residues, 112 versus 158, we have these different polymorphisms that are associated with this. So ApoE3 is where we would have a cysteine and an arginine. And ApoE4 is where we actually have two different arginines. And it turns out that when you look at the risk factor of potentially developing Alzheimer's disease, so imagine everything else being equal, so you wipe out all the known risk factors that are environmental, so not exercising, having like no strokes, etc., maintaining your cholesterol levels overall, and you have this sort of in your background. If you look at an individual who has an ApoE2 status versus ApoE4, everything else being equal, and everything else in the genetics being equal, the ApoE4 individual, if they have two copies, they're more likely to develop late-stage Alzheimer's disease. So again, it's the only identified risk factor that seems to be consistent within the literature. And we would have these two arginines. And ApoE2 actually tends to be protected, so you will be less likely, everything else being equal, to develop Alzheimer's disease later on in life. There are some trade-offs because of the way that cholesterol is important in other parts of the body. So even though it's protected, ApoE2 is actually more likely to cause you to die of a heart attack than for you to die of something related to Alzheimer's disease. So there are some trade-offs there. And so the take-home message is always be normal. And so the ApoE3 is your best option. Don't try to be like over or extra, or don't try to be like under. Just try to be normal. Normal is nice. So this ApoE3 is actually the best choice. So why talk about this? So these ApoE molecules involved in cholesterol processing and transport, associated with microglial cells and astrocytes, are important also in being able to bind to amyloid beta. And amyloid beta will bind to these different ApoE isoforms. And it turns out that if we lower ApoEs in general, this actually causes an increase in beta amyloid deposition. And if we look at the correlation, the ones that actually clear or don't clear as well or as efficiently are microglia that contain the ApoE4 isoform. So more and more of the amyloid beta will build up. Normally this is there to transport cholesterol, but it can also transport amyloid beta. If you're producing it and not being able to clear it, it might actually lead to the cytotoxicity that we've been talking about so far. So this ApoE isoform seems to be really important, but in addition to that, how it gets itself lipidated or activated through lipidation actually determines how active it will be. And some of this goes back into what lipidation happens within the brain, and this occurs through this ABC1 alpha, or alpha 1, and this is an ATP binding consent transport molecule that will change the lipidation status of ApoE. So let's talk about this in a little bit more detail. So a couple of things to keep in mind. So this is sort of, even though I have a few more slides to talk about related to, since nothing works, let's talk about something that might work, and let's talk about something that might work also in humans, not just mice, because mice models are so boring, etc. They don't really work. They are maybe useful, maybe not. So let's bypass those and talk about a therapy here in Toronto that actually seems to work really well for Alzheimer's. And then before we get there, let's talk about what you do need to know. So these next few slides, and the ones that deal with the overall role of ApoE in microglial cells and the processing of amyloid beta, these next few slides are going to be important. And the slides that come after, you really don't have to worry about being on the final exam at all. It's just to get you interested in Alzheimer's and what is happening here in Toronto. So again, if we're taking a look, we want to be able to have the amyloid beta, and we need to be able to break down amyloid beta. We need to clear it as it is being formed. So we can form it, beta secretase is involved in its processing, amyloid precursor protein is on every cell, and we need a way to be able to clear this amyloid beta, and we need to get it out of the brain. One possibility is to simply look at these microglial cells as a way in which these amyloid beta composites can actually be eliminated. So there are a couple of things that this can actually do. So these microglial cells and the lipidated or activated forms of ApoE, and there are different isoforms, and these isoforms are represented here by this sort of gold oval surrounded by the two orange-red bars. One of the things that we know is different forms of ApoE, ApoE 2, 3, and 4, can help and activate the breakdown process overall. And one of the ways in which it does this is that ApoE can actually be released or be part of this complex known as insulin-degrading enzyme. And this, again, is just as you would suspect. It involves insulin. We need to get rid of insulin within the brain. And this degrading enzyme gets activated by this lipidated form of ApoE. And one of the things that this allows it to do is to process and break down the beta amyloid that happens to get formed. So that's one process by which this can occur. Another process that allows this to happen is that when we get these microglial cells, we also have this ApoE, and it's lipidated ApoE, and it can bind to this amyloid beta. So that was the previous slide talking about how different ApoE isoforms can bind to amyloid beta better than others. E4 is the worst. It doesn't bind very well. It doesn't clear it very well. ApoE 2 is the best, but we're really hoping for ApoE 3. And the thing that happens on microglial cells is that with this lipidated ApoE, we can also bind it and cause it, this entire complex, to be internalized, because on the surface of the microglial cells, we have this molecule known as neprilysin. And neprilysin, together with this ApoE, lipidated ApoE, plus the amyloid beta, as a complex, will be internalized within the cell. And within the cell, this entire complex, including the amyloid beta, which it has bound, and some lipidated ApoE binds better than others, will be broken down within the endosomes and lysosomes of the microglial cells. And this neprilysin is a good marker, because think a little bit about how we have approached and strategically approached a number of different cells or neurons, and how we've tried to identify specific markers for these different cells or different types of neurons. And neprilysin is one of those markers that seems to be important and localized to these microglial cells. And so we need to consider that breakdown process as well. So again, here we're looking specifically within the microglia, and I've just taken you through this entire process through these microglial cells. I'm going to tell you that you don't need to worry about this LXR pathway, RXR pathway. What I've just covered is pretty much it for what you need to know for your final exam. So at this point, you can just sort of cruise, unless you have questions. So I haven't actually taken the time up until now to ask for any questions. So if you have any, let me know. This pathway here on the right, you don't need to worry about. For the final exam, everything up until this point in time, you need to know. And there will be pretty good coverage from today's lecture. But do you have any questions from anything from lecture so far? Because that's pretty much it. But still show up for Wednesday. No? Okay, so if not, and we won't go through this in any more detail. So a lot of these different drugs, and you don't have to really worry about any of the things here. And you don't have to worry about this scanning ultrasound technology. I just wanted you to be aware of this because it's actually here in Toronto, and it's something that is being researched here in Toronto. And even better, it's not just being researched in mice, it's actually being researched in humans. And it's really one of these developing therapies that seems to be really, really cool and really, really effective. So a few years back, it's only been about 4 years now, so back in 2015, there were all these papers that were suddenly coming out saying that, hey, you know what? We don't have therapies for Alzheimer's disease. And back in 2015, they were even predicting we probably won't have any therapies for Alzheimer's disease. And guess what? They were right. And so they needed to think a little bit about how do we get rid of these amyloid beta plaques, and can we actually improve Alzheimer's disease, and this gets into this animal model of Alzheimer's disease. And one of the things that we need to understand about ultrasound is that ultrasound uses these sound waves, as you would probably guess. And these sound waves have a variety of different interesting properties, but one of the things that it can actually do is you can actually use that sound wave, compress it, expand it, and this allows you to get this sort of echo effect. And this means that a molecule, like air, can be compressed back and forth, and that's how you hear my voice right now. So my voice is causing the different air molecules to vibrate at a certain frequency, and if we had a molecule that was put into your bloodstream and we had an ultrasound source, it would also cause that molecule to vibrate at a certain frequency. And so one of the first things that people wanted to know is if we put in like an inert gas, we have these little inert gas molecules, and we package them in a way that we could deliver into the brain, could we open up this blood-brain barrier? Because all the drugs that have been used up until now, the biggest issue with these Alzheimer's disease drugs and even stroke and other types of drugs, this blood-brain barrier actually prevents many of the drugs from getting into the neurons, into the brain. So could we break open the blood-brain barrier? That was sort of the big sort of wish. And so it turns out that if you look at different types of drugs and you looked at their delivery after breaking the blood-brain barrier open, that you could actually enhance the delivery of different drugs. And so this goes back into, and again, no need to worry about any of the details here. If we look at even the delivery of adenoviruses past the blood-brain barrier, without like doing stereotactic injections, et cetera, this is sort of the future of what we think is going to happen for delivery of viruses to the brain. Would we be able to use focused ultrasound to break open these tight junction barriers to allow for the delivery of different viruses and different particles? And might we be able to activate different cell types as a result? So it turns out that yes, we can do this, and it actually works incredibly well. And the original thought and the original idea was, hey, let's do this so we can get more drugs that might treat Alzheimer's into the brain, and let's make the brain better that way. And that was sort of the idea. But the thing was really, really weird. When we do those experiments, when we treat the animal model that has Alzheimer's disease, and we also treat the control animals, and we are delivering drugs, and we're just delivering the control vehicle, the control animals actually, after focused ultrasound, also improved a whole lot. And you kind of wondered, like, hey, there's something weird. So either our results are wrong, because we talk about results not being correct and accurate, or there's something else going on. And it turned out that it's not just the drugs, it's actually breaking open the blood-brain barrier that seemed to be really, really effective in Alzheimer's disease. And it was one of those really interesting things that people were looking at. And again, you have to think about this, and you have to think about it this way. And when you talk to neurosurgeons who are doing this here in Toronto, one of the biggest concerns is, okay, you open the blood-brain barrier, does it fix itself after a period of time? How many of you think that it does? It goes back to normal? A couple of you, brave people at the back. Thank you, brave people at the back. And it does, and it turns out that you can break open the blood-brain barrier, and then it will heal itself after a certain amount of time. What's the other caveat, do you think? So let's think like clinicians for a second. So you can break open the blood-brain barrier. It can repair itself. Who are normally involved in these studies? Do you look at older individuals or younger individuals? Older? So if you were to walk around campus today and you're looking at participants needed, $15 an hour, who are they targeting? They're targeting you, your age, and so young brains, blood-brain barriers open, they can close. But that's not who's normally affected by Alzheimer's disease, and that was one of the big concerns is, can we actually cause the brain, in older individuals, the blood-brain barrier to reform again? And it turns out that in trial after trial that's been done at Sunnybrook Hospital, as well as in the United States and in Europe, that older brains too can also have their blood-brain barriers coming back intact. And so this study was actually the first study to show that all you have to do to treat Alzheimer's is to open the blood-brain barrier. And if you open the blood-brain barrier for a very short period of time, the brain will allow itself to be fixed. And it was kind of a remarkable story that needed a lot of proof to show that, one, the blood-brain barrier actually could be opened, and it was very clear that it could, and that you could actually see the movement of cells into the area of the brain past the blood-brain barrier being opened, and it was actually a really, really cool study. And you could see that this dye that normally cannot penetrate the blood-brain barrier, this blue dye known as trypan blue, it's normally excluded from the brain. So if you inject a mouse with trypan blue, the whole mouse will turn blue, its tail will turn blue, its feet will turn blue, most of its skin will turn blue, but its brain is usually protected, except when you open the blood-brain barrier, as you have seen here, and then the brain gets flooded with this trypan blue, showing that the blood-brain barrier does get open. Again, no big deal. But one of the things that is really cool about this is that when you look at the brain of these mice that have like Alzheimer's disease plaques, by breaking open the blood-brain barrier, you can actually make the mouse better. You can actually reduce the plaques. You can actually get rid of a lot of the beta-amyloid, and there's no cell death. Breaking open the blood-brain barrier was thought to be really, really bad. People were thinking, if you do that, you're going to like destroy the brain's not going to survive, but it turns out it's not a bad thing if it's done for very short periods of time. And again, there was no cell death that was shown, and again, really remarkable, and what we're looking at is just a controlled, you put the little ultrasound probe on top of the brain, but you don't turn it on, and then the other animal, you do turn on the ultrasound, and you can see that the number of plaques actually significantly decreases. The animal has better memory, etc. And this is done in mice. But what's more important and what's more exciting is that these early studies back in 2015 actually led to a number of humans, small human clinical trials. And these small human clinical trials that have been started here in Toronto have shown that there is some improvement. And if you were to make a guess, or you can jump to the very end of your notes, what do you think ultimately breaking open the blood-brain barrier does to make the brain fix itself? If you were to guess. Guess. Sorry? It releases more A beta. And then what? So how does the brain fix itself? What do you think also gets activated in this process? Guess. Yes. The immune system. What particular type of cells do you think get activated after focused ultrasound or sustained or scanning ultrasound? What immune cells in the brain? Sorry? The microglial cells. And it turns out that just by activating the microglial cells for a very short period of time is enough to help with the clearance, at least in this animal study, and in repeated sort of blood-brain barrier openings at Sunnybrook. And if you happen to take some fourth-year seminar courses, either in LMP or in other, or PSL, you'll hear about Dr. Lipsman, and you'll hear a little bit about the things that he does in more detail. And it's actually really exciting because we don't have drugs that work, we don't have therapies that work, but this seems to be kind of effective, and it might be working through a biological process that I hope now you might be able to understand a little bit more through those microglial cells? So that's a great question too, but we don't know. Like we would ideally, now that we have markers for different types of microglial cells, so we know TSPO, which is a translocator protein associated with some microglial cells, can identify activated versus non-activated microglial cells. It's a great question. They don't have that data. I believe that pro-inflammatory would cause a lot more problems, but again, they don't have that data, and it would be pure speculation. It would be a good hypothesis to go and test. Any other questions? No? Yes? How many of you are really worried for the final exam? Oh, okay. Well, that's very intimidating, isn't it? So I think you should always be a little bit concerned about the final exam. And on Wednesday, I'm going to take you through the different parts of the final exam, how you might get ready for the final exam. We have not had a lot of success streaming in the MedSci auditorium, so we're not even going to try, but you can record and do whatever. And how many of you would like some questions in advance of the final exam? Not everyone. Okay, not everyone. Surprise. Do you guys really like surprises? No? No? Okay. So shall I ask again? How many of you would like a couple of questions in advance? Again, not everyone's hand is going up. All right, okay, we will see on Wednesday. Until Wednesday, have a great week. Try to stay well as much as you can. You don't have to worry about any of this material, and just it's for your own interest.